Good morning, everyone. Can you hear me clearly? Excellent. I think we can start now. It's a great pleasure to be here. My name is Maciej Olek, and I represent the Evonik company. For today, I've prepared what I believe is a very interesting topic, as I'm combining two quite significant trends from the cosmetic market into one presentation. So I'll be talking about exosomes and also collagen. And all of this will be presented as a kind of duet that works together in harmony. To begin with, I'd like to start by presenting our brand new product line, which we've named Vicolage Fortify. We're talking here about products that are, it's a vegan collagen, meaning collagen that we specifically obtain from plants through fermentation processes which are biotechnological. And first and foremost, we are talking here about collagen, which is identical to that which naturally occurs in our skin. It is type 3 collagen, the one that naturally occurs in human skin. A very interesting property of this product is precisely its connection with exosomes, and I will elaborate on this in the later part of this presentation. And above all else, this particular product line is incredibly effective. We have some truly fantastic clinical trial results right here, which I would like to briefly present to all of you today before we move on to discuss the subject of exosomes. Currently, within our extensive Vicolage Fortify product line, we present two specific products. They are, in their essence, remarkably similar to one another. Both of these formulations, as you can easily perceive from their INSI ingredient list, contain polypeptide 69 at exactly the same concentration. The most significant difference in the composition of these two products is that one includes additional ingredients which are preservatives, while the other, this GP, is completely free of preservatives, meaning it contains no preservatives at all. As you can see from the INSI list, this particular product contains a polypeptide. Its name is polypeptide 69, not collagen. You are certainly aware that PCPC has specifically reserved the INSI name collagen, that exact term on the INSI list. The PEC for collagen of animal origin. So what we have here is vegan collagen. However, we're not allowed to use the specific term collagen or its English equivalent in NC. As I mentioned, we're talking here about a product that is a one-to-one reflection of the type 3 collagen chain, the one found in human skin, meaning a natural component of our dermis, our true skin. We're using an amino acid sequence that reflects about 50% of this chain. Why not 100%? These molecules would be too large. They would have trouble penetrating our skin. And I'll talk a little more about penetration later. Therefore, we made the decision here to implement this cut, reducing it to 50% of the total chain's length. This means we now possess a particle that remains quite substantial, measuring 50 kilodaltons. And this is the active ingredient, polypeptide 69, that we have here. This product is truly incredibly effective and works on multiple levels. Here we've schematically illustrated how it enhances the components of the so-called dermal-epidermal junction, which is the crucial connection between the epidermis and the dermis. It remarkably boosts the components of our collagenous protein matrix, including both elastin and fibronectin, but it also possesses protective functions. So it's also capable of reducing those factors which degrade proteins and also degrade collagen within our skin. And perhaps just briefly, I'll share a few results from our own studies. These particular findings are ex vivo, meaning we utilized explants, correct, from donors, which means actual human skin. And we simply proceeded to examine the increase or upregulation of various types of proteins, or indeed the markers associated with these proteins present in our skin. The results are truly fantastic because of a 250% increase in pro-collagen type 1, 30% of collagen type 3, and so on and so forth. Also a fantastic enhancement of elastin, fibronectin, and the cross-linker, that crucial protein responsible for maintaining the entire network of elastin within the connective tissue matrix of our skin. We also got some really great results, but we've also already tested this here in vivo studies on different areas of our own faces or the faces of the participants in this particular study. As you can see, we had 35 volunteers, which was quite a substantial research sample. We focused on different facial areas and conducted a clinical dermatological study to assess the reduction in the number of wrinkles. And the results here are truly very impressive, and various parts of our face, this lifting, this firming of the skin is also clearly reflected in the reduction of wrinkles. So it's very nicely visible here. Also in another study where we utilized a different technology, meaning not a dermatological assessment, but also a visual evaluation. And here it's also clearly visible, especially on these contrasting images, that this reduction is indeed very significant. And naturally, when we boost collagen, we expect the skin to be stronger, more resilient, and that it also gains elasticity thanks to this elastin boost. And these results, which we obtained in this in vivo study, also confirm this. Well, an incredible 20% increase in the firmness of the skin, which means a strengthening of the skin itself, thanks to collagen, plus almost 20% more elasticity for our skin. And also very interesting results regarding the skin's density. Well, since we're getting more proteins in the dermal layer, we naturally expect that the density of the skin will also be significantly strengthened. And these results truly confirm it, a 30% increase. This is an incredibly high value when it comes to the density of our skin. I would also like to briefly present to you the very latest research that we have recently conducted. Here we are discussing aesthetic medicine procedures and the specific application of our collagen as a crucial preparation for the skin before such a treatment, as well as its beneficial use after these procedures to effectively strengthen, improve, and accelerate skin regeneration. We're talking about microneedling, which is microneedling. We're discussing laser treatments and also laser procedures. We're also talking about chemical peels. These studies were conducted on a significant group of 64 participants. And of course, aesthetic medical procedures, the ones we've presented here, which we utilized in these clinical studies, are, well, invasive, meaning we're talking about a certain degree of skin damage. And that's why we used Vicolage to improve and strengthen the regeneration of the skin and its return to a proper and healthy state after quite aggressive and invasive aesthetic procedures. I'll be very brief here, as I don't have the time to delve into every single result that we've obtained, but we were indeed able to observe a statistically significant improvement in terms of skin quality and its regeneration following these aesthetic medicine treatments. For example, skin density, its improvement, and enhanced skin hydration. And what's very interesting, these skin discolorations were also significantly reduced by 40% and at a faster rate, as it's well known that skin after such treatments is quite severely stressed. All right then, Mr. Maciej Olek from Evonik spoke for almost 10 minutes about Vicolage, but where are those exosomes? So now let's talk about exosomes. And here I've prepared a rather simple graphic, but I believe I'll be able to explain the role that exosomes play in the context of this product that I've just been telling you about. Of course, you are all familiar with the cross-section of the skin. It is also a well-established fact that in this epidermal layer, the primary cells are keratinocytes. We also have fibroblasts. Not only that, we possess many more skin cells, but let's concentrate on these two key elements. And now we proceed to apply the product known as Collagen 45 directly onto the skin's surface. So we're talking about our polypeptide 69, which is type 3 collagen. These are quite substantial molecules, 50 kilodaltons in size. The penetration of such molecules is quite significantly limited. Generally, we are aware that smaller molecules, those under half a kilodalton, readily penetrate our skin. Naturally, there are specific conditions and various factors that can either enhance or diminish this effect. However, in our case, we are dealing with a substantial molecule of 50 kilodaltons. Yet, despite its size, our in vivo research ultimately demonstrated that our product does indeed function effectively. It simply cannot function effectively on the surface. It must be actively working deep within our skin. That's why we conducted a series of diverse studies to uncover precisely what happens, that under the influence of our product, our skin gains more collagen, more elastin, becomes significantly strengthened, firmed, and facial wrinkles are visibly reduced. Here, the microbiome comes to our rescue, that is, these billions of tiny organisms living on our skin, which essentially perform a vital function for us. Microbiomes, of course, have a natural tendency to expand their colonies. These are bacteria, various types of microorganisms. They require sustenance. They release enzymes and various kinds of particles that are found on our skin, which they then use for their sustenance. So, enzymatically, they possess the capability to metabolize, or to put it in simpler terms, to break down and cut up bigger structures into much smaller fragments, such as our 50 kilodalton polypeptide. And this process actually takes place. These smaller fragments, in turn, then gain the ability to easily penetrate the skin. And what happens from that point onwards? We observed that overall, these smaller fragments of our collagen actually interact with keratinocytes. These keratinocytes, they provide a certain kind of information, keratinocytes, that something bad is happening to my neighbor below, to the fibroblasts, because collagen fragments in the epidermis, in the skin, that is not something normal. This means I have to send information to my neighbor below, to the fibroblasts, that something is not quite right on the floor, directly beneath. And indeed, this truly happens. This interaction takes place, and keratinocytes send a specific kind of messenger, like WhatsApp or an SMS. They simply transmit information in the form of exosomes directly to fibroblasts. Exosomes are a specific type of messenger, a communicator between skin cells, and within these exosomes, information is contained, information that is genetically or molecularly encoded, which is then transferred to fibroblasts. So fibroblasts then read this information, and in response, for example, they begin to produce more collagen or various other types of proteins. What I've just told you, we refer to this as epigenetic regulation, where exosomes play a very specific mediating role between the skin cells. What I have just presented to you is not merely a hypothetical situation. Indeed, to confirm this mechanism that I have just shown you, we have a whole series of interesting studies. I don't have time to go into the specifics of these studies, but we did actually examine this at the level of skin microbiome metabolism, and we observed that both under aerobic and anaerobic conditions, these molecules are broken down, metabolized, essentially cut into smaller fragments. In an in vivo study, we observed that indeed these smaller fragments do penetrate the skin, which means after applying our 50 kilodalton Collagen 45, this penetration doesn't happen right away because these enzymes need to act to cut it into smaller fragments. However, in our in vivo studies, we have observed that these fragments effectively penetrate the skin. Subsequently, we were able to isolate the exosomes that are produced by keratinocytes and analyze them using the PCR method, correct? So, we were investigating whether there's any genetic information encoded for fibroblasts, and we actually discovered it. We also have a series of various results here, which indeed demonstrate that this signal, this information, is transmitted to fibroblasts. And what's interesting, we also have studies that show that fibroblasts, which were treated with these exosomes, properly decode this information and react accordingly. In this study, fibroblasts completely didn't perceive our product. Only keratinocytes were subjected to the influence of our product. And we then isolated the exosomes that were produced by these keratinocytes. And the fibroblasts only saw these exosomes, meaning the information encoded within those exosomes. And indeed, our clinical studies confirm that it works, that there's a truly remarkable improvement here. But I already showed you that in the first part of my presentation. This is also a very interesting product, yielding fantastic results. And also very interesting is this concept of epigenetic regulation, where exosomes actually play the role of a unique mediator, a communicator between skin cells. So that concludes my presentation for this moment. If you have any questions, I am at your full disposal right after this talk or at booth 430. I invite you and wish you a very pleasant day. Thank you.